Assalamu alaikum. Thank you everyone for being connected with me. Today is my second class on pharmacodynamics series. So in my previous class I have discussed about the drug targets and basic conception on pharmacodynamics. Today I am going to about receptors to discuss in details. So I just review for brief time. So I have mentioned that according to activity, there are three types of receptor: active receptor, spare receptor, silent receptor. Okay. Now today is our topics is according to mechanism of action. According to mechanism of action, the receptors are of mainly four types. That means ion channel coupled receptor, G protein coupled receptor, tyrosine kinase attached or any other enzyme attached receptor, and number 4 one is DNA coupled receptor. So I will describe everything in details inshallah. So at first come to the point, ion channel coupled receptor. So what have you understand from these headings? That means the receptor is attached or connected with an ion channel. So look at the two classical examples of these types of receptor, such as nicotinic receptor. So what is the agonist for the nicotinic receptors? You know that acetylcholine. So look at the picture. Here you can see actually a channel-like structure, a channel or a tunnel. Okay. So in this tunnel, which ions is transmitting? It is called sodium ion. That means through this channel or tunnel, sodium ion enter into the cell from outside of the cell. Enter into the cell. Okay. So this channel is remain closed. So when will it open? It will open after binding with the acetylcholine with its receptor. That means when acetylcholine attached with the receptor, then this receptor will open this sodium channel, and after opening, the sodium ion will able to enter into the cell from the outside. So whenever the sodium begin to enter into the cell, so what will happen? As the sodium is an positive ions, that's why the positive polar ion will be increasing inside of the cell. So what will happen in next step? That means action potential will happen in the cell, because we know that all of you know that the graph of action potential, then repolarization. That means it is called simply depolarization, and depolarization happen when there will be entrance of sodium ion. And when will be repolarization? This phase is repolarization. Then means the membrane potential come towards its basal level. So it is called repolarization. So when will repolarization happen? When potassium ion will be effluxed, or when potassium ion will come out from the cell. Okay, that means when potassium ion will leave the cell. Okay, so this is called repolarization, because if potassium ion leaves the cell, that means there will be negative polarity inside the cell, because the positive ion is leaving the cell. So negative charge will be increasing inside the cell. Okay, so this is called repolarization simply. That means we have shown you that the sodium channel remain closed, but it will open when acetylcholine bind with the receptor. So this receptor shows its effect by opening sodium channel. That's why this receptor is called ion channel coupled receptor. Okay, I hope all of you understood. inshallah. So look at the next example, most common example it is also. This is GABA receptor. GABA receptor. So what have you looked there? You can see here a channel like structure, just like as previous, a channel like structure, and this channel also has some subunit. Here you can see some subunit of this sodium channel. That means alpha subunit, gamma subunit, delta subunit, again alpha subunit, beta subunit. Okay, beta subunit. Now look at the GABA receptor. It has also these types of subunit such as alpha subunit, beta subunit, alpha subunit, beta subunit, gamma subunit. So between the, remember it, between the alpha subunit and gamma subunit, there is receptor for benzodiazepine agonist. So the benzodiazepine binds with its receptor, which is located between the alpha subunit and gamma subunit of GABA receptor. That means this is actually GABA receptor, and benzodiazepine binds with a specific location of this GABA receptor. So what will happen after binding this benzodiazepine? When benzodiazepine bind with its receptor, the affinity of the receptor towards GABA will increase. That means now after binding of benzodiazepine, the GABA will more and more attached with this GABA receptor, more and more will attached. So what will happen? That means now GABA agonist more and more will attached with its receptor. So all of you know that when GABA attached with GABA receptor, this channel will be opened. That means this channel is just now closed. So after binding of GABA with its receptor, this channel will be opened, and which ions can transmit through this channel? This is chloride. This is chloride ion. So you know that the chloride ion concentration is more in extracellular space. That means here are more chloride in extracellular space, because this is cell membrane. Okay, and this is the cytoplasm. So when this chloride channel will be opened after binding of GABA with GABA receptor, then all of this chloride ion will enter into the cell. They will begin to enter into the cell. So what will happen? As a negative ion, that means as anions is entering into the cell, that's why there will be increment of negative charge inside the cell. So it is called hyperpolarization. So you have understood that this GABA receptor will play a inhibitory effect on cell, because this GABA receptor is causing hyperpolarization to the cell. So if a cell become hyperpolarized, so the action potential cannot be happen in that cell. Okay, so benzodiazepine is causing increasing, that means benzodiazepine is increasing the affinity of the GABA receptor to its ligand GABA. That's why the neuron or the effector cell become suppressed. Why? Because the effector cell is becoming hyperpolarized due to this effect. Okay, so this is another example. Now come to the most important and a little bit complex protein coupled receptor, that means G protein coupled receptor. That means these receptors are attached with a specific protein which is known as G protein. That means this receptor is attached with G protein. So there are many receptors, that is G protein coupled receptor. I have written there just three important example: adrenergic, cholinergic receptors and histaminergic receptors. So look at the structure of G protein coupled receptor at first. This is a figure from Kerchang. So look at the picture. This is the cell membrane and this is the receptors. This chain-like structure is receptors. And look at right side, the G protein is attached with a portion of this receptor. G protein is attached with a portion of this receptor. That means this is a receptor, and here is another important information. This receptor traverses the cell membrane for 7 times. It traverses or pierces the cell membrane for 7 times. Look at the figure. First time, second time, third time, fourth time, fifth time, sixth time, seventh time. Then it is terminal. That means this receptor, G protein coupled receptor, traverses the cell membrane for 7 times, and the initial end is called N-terminal or amino terminal, and the terminal end of this receptor is called carboxy terminal. Okay, because this terminal is containing amino residues and this terminal containing carboxylic residues. Okay, so when the agonist will bind with this receptor, so this agonist bind with the receptor between first and second arm. Look at the figure. The agonist is bind with the receptor just between the first and second arm. Okay, so after binding with its agonist, the receptor will be activated. The receptor will be activated and a activation signal will transmit throughout the receptors, and this signal is transmitting towards the inside of cell, and it will now activated the attached G protein. That means now G protein is activated. That means now G protein is activated. Now what will happen? This G protein will activate some enzyme. Okay, so what enzyme will be activated? It depends on the nature of G protein. I will discuss it just later, inshallah. So look, so here you have seen that the structure of G protein. Okay, and the G protein is attached with these receptors. That means between the, look at the arm number, this G protein is attached with arm 5 and arm 6 of this receptor. Arm 5 and arm 6 of this receptor. In this loop, G protein is attached. Okay, now come to the next point. This is a chart from Predjan. If you can memorize it, you can explain every G protein coupled receptor mechanism of action, inshallah. So all the information isn't needed for you. You just remember GS, GI, GQ. Okay, so GS protein activates adenylyl cyclase enzyme, GI protein inhibit adenylyl cyclase enzyme, GQ protein activate phospholipase C enzyme. So that means GS protein activates adenylyl cyclase enzyme, and this adenylyl cyclase enzyme will form cyclin AMP from which? From ATP. That means adenylyl cyclase will increase intracellular cyclin AMP level because it is the function of this adenylyl cyclase enzyme. And as GI protein suppressing the adenylyl cyclase enzyme, that's why cyclin AMP level will be decreased, because the GI protein inhibiting the adenylyl cyclase enzyme. So cyclin AMP level will be decreased inside the cell. And next one is GQ coupled protein. GQ protein will activate phospholipase C enzyme, and this phospholipase C enzyme will form inositol triphosphate and diacylglycerol. That means this phospholipase C enzyme form these two residue from which substrate? It is called phosphatidyl inositol biphosphate. That means phospholipase C break down the phosphatidyl inositol biphosphate and will form inositol triphosphate and diacylglycerol. This inositol triphosphate then will increase the intracellular calcium concentration by opening calcium channel in the cell membrane. Okay, so this increased intracellular calcium concentration will show the effects. So this is all about the G protein coupled receptor. Now look at the flow chart about the G protein coupled receptor. So number first is GS. So this is the cell membrane and here GS protein is attached, and this is the G protein coupled receptor, and this is the agonist of the receptor. Suppose this agonist is norepinephrine. So norepinephrine binds with the receptor. Now the receptor will be activated. And G protein will be activated. Now this G protein is GS types. GS is stimulatory. So I have told you that GS protein activates adenylyl cyclase. So adenylyl cyclase will be activated more and more. So this more and more activated adenylyl cyclase will form more and more cyclin AMP. So intracellular cyclin AMP level will increase. So this increased cyclin AMP, then this increased cyclin AMP will form more and more cyclin AMP dependent protein kinase. That means there is inactivated cyclin AMP dependent protein kinase enzyme, and when this enzyme will get more and more cyclin AMP, then this cyclin AMP dependent enzyme become activated. That means this is the activated form of cyclin AMP dependent enzyme. So this cyclin AMP dependent activated enzyme now will activate more protein into activated form. That means this enzyme now will activate more protein. So here I want to give you information that whenever phosphate will attach with any substrate, suppose this S is now single and now phosphate is bound with it. That means, so this S is now activated, or this protein is now activated. So look at the picture. That means increased cyclin AMP will activate the cyclin AMP dependent protein kinase. So this activated cyclin AMP dependent protein kinase now will activate more and more protein into their active form, and this active form protein will show the effect. Okay, and if it is GI, just everything will be reversed. GI protein, that means when agonist will bind with the receptor will activate GI protein. So adenylyl cyclase will be suppressed. GI means inhibitory. GI means inhibitory. So adenylyl cyclase will be inhibited. So cyclin AMP will be formed in decreased amount. So cyclin AMP dependent kinase protein will not be activated adequately. Because cyclin AMP is now not available. So this activation of this protein, intracellular protein, will not happen. So response will not be happened. That means there will be no response. So GI protein will show a inhibitory effect on cell and GS protein will show excitatory effect on cell. Now next one is GQ coupled protein. So this is the receptor and this receptor is attached with GQ protein. So whenever the agonist will bind with the receptor, then GQ protein will be activated, and I have told you that GQ protein activates phospholipase C. This phospholipase C then break down the phosphatidyl inositol biphosphate into inositol triphosphate and diacylglycerol. Okay, now this inositol triphosphate will release more and more calcium from the sarcoplasmic reticulum. This is called the sarcoplasmic reticulum, and this inositol triphosphate also open more and more calcium channel in cell membrane. That's why more and more calcium will begin to enter into the cell from extracellular space. That's why overall intracellular calcium concentration will be rise. Okay, and in another situation, the form diacylglycerol, this will activate the protein kinase C enzyme. This diacylglycerol will activate the protein kinase C enzyme. This protein kinase C enzyme will activate more and more protein into their activated form. Look at the figure, just like the previous explanation. This S was single and phosphate is added, now it become SP. That means S is inactivated form and SP is activated form. So this activated protein will show its response. Okay, so this is all about the 3 G protein coupled receptor mechanism. These are the main G protein coupled receptors. Okay, now we can again classify the receptors according to onset of action. Within millisecond, there are nicotinic receptors, GABA receptor. That means these two receptors shows its effects very rapidly, and within seconds, the onset of action. Okay, onset of action, not duration. Remember, that means the time required to show the effect. Okay, within second, this is adrenergic and cholinergic. That means after binding of the agonist with these receptors, you will find the effect after 1 second. That means 1 second will be consumed to show you the response. Within minutes, that means there is more time required. Insulin receptor, growth factor receptor, cytokine receptor. So there is the onset is little bit slow than the previous one. Within hours, it is more slow receptors. What are these receptors? Steroid receptors, thyroid receptor. Why these receptors are more slow? That means why more time is needed to show the response? Because these receptors goes to the nucleus, then they bind with the DNA, then they will do the transcription, formation of mRNA, then this mRNA come outside of the nucleus, then this mRNA will be translated into a specific protein, and this protein will show the response. That's why more time is required for these receptors.